Hello. So I decided to focus this research project on Rett syndrome. Out of the list given to us for the week 9 discussion, I knew very little about Rett syndrome and was intrigued. So here we go. I'll start with a brief history of Rett syndrome. Observations were first made way back in 1954 by Dr. Andreas Rett. He first compared the clinical and developmental histories of two girls who he observed making similar repetitive hand motions and found many similarities. Rett traveled around Europe in search for more similar individuals. He later published his findings in German medical journals around 1966. In 1960, Dr. Bengt Hagberg began to observe similar behaviors in some young girls and wrote an article on Rett syndrome in 1982, raising the profile of the syndrome and bringing awareness to it. A major breakthrough occurred in 1999 when Ruthie Amir, a researcher at Baylor College of Medicine, discovered MECP2. The mutation of MECP2 is what causes Rett syndrome, which is something I'll discuss more in depth in a few slides. What is Rett syndrome? Rett syndrome is an extremely rare brain disorder that is almost exclusively found in females, with about 1 in 9,000 to 10,000 females being affected. There's a large variety of symptoms that come with Rett syndrome, but the severity is determined by the location, type, and severity of the mutation to the gene, resulting in every individual with Rett syndrome looking different with different symptoms. Individuals with Rett syndrome experience anywhere from 6 to 18 months of standard development before symptoms begin to arise. These symptoms can include loss of purposeful hand function, replaced with repetitive, distinct hand motions such as ringing or clapping, cognitive issues, including learning, communication, speech, and coordination difficulties. Physical growth is slowed, muscle weakness and stiffness occurs, abnormal walking patterns, such as too wide of a gait or only walking on the toes, breathing difficulties, excessive drooling or spitting, unusual eye movements, irritability, and seizures. Methods of treatment are in development, and symptoms can be treated with a variety of medications, surgeries, and physical therapy. Causes and inheritance. Rett syndrome is caused by mutations within the methyl CpG binding protein 2, MECP2 for short, gene, which is responsible for creating the MECP2 protein that set out to regulate gene activity and brain function. In most cases, Rett syndrome, there is no family history of the mutation. In the few cases where there is more than one affected family member, the MECP2 gene mutation have an X-linked dominant pattern of inheritance. A little more about methyl CpG binding protein 2. Its official symbol is MECP2. The gene type is protein coding, and the organism is Homo sapiens, also known as humans. DNA methylation plays an essential role in human development. MECP2, MBD1, MBD2, MBD3, and MBD4 comprise a family of nuclear proteins related by the presence in each of a methyl CpG binding domain. MECP2 is X-linked and essential for embryonic development. Mutations are the cause of most cases of Rett syndrome and is one of the most common causes of cognitive disability in females. MECP2 is expressed in 27 different kinds of tissue, including the ovary, brain, kidney, lung, prostate, and thyroid, to name a few. The genomic location is XQ28. The exon count is 12, and the protein length is 498AA. As for patient support and advocacy resources for Rett syndrome, the National Organization for Rare Diseases, also known as NORD, is a great site to check out. NORD provides support to individuals diagnosed with rare disorders, caregivers, and advancing research on rare disorders. The NORD Rare Disease Database is a great source when researching. A quick search pulls up articles directly created by NORD along with outside sources that can provide even more information. I included a screenshot of the site's menu bar in this slide just to really show the amount of information NORD is providing here. You can understand rare diseases. You can understand what it's like living with a rare disease. A lot of individuals who are living with a rare disease share their stories on this site. You can provide community support, you can receive community support, you can advance research, and you can get involved, which includes attending upcoming events put on by NORD. You can volunteer, intern, and even find a career with NORD, and of course, you can donate to their cause. I found a few clinical trials on Rett syndrome. The first one investigates the safety of using NNZ-2566, also known as trofinetide, as a treatment method for Rett syndrome. The next trial investigates sleep patterns in individuals with Rett syndrome, with the goal of identifying sleep disorders in affected individuals compared to children who are not affected, and how disordered sleep affect the everyday behavior of individuals with Rett syndrome. Another trial searches to combine the gene editing ability of CRISPR/Cas9 and gene therapy from the adeno-associated virus, AAV-based delivery technique to correct mutations in MECP2. The first research article I decided to focus on, titled Therapeutic Effects of Extracorporeal Shock Wave Therapy on Patients with Spastic Cerebral Palsy and Rett Syndrome, tested the effects of extracorporeal shock wave therapy, also known as ESWT, on children with Rett syndrome. ESWT has previously been used on children with cerebral palsy to help improve motor function and muscle stiffness. So since the symptoms of Rett syndrome are so similar to cerebral palsy, they wanted to test and see if this would be helpful for them as well. The results of the study did show clinical improvement of spasticity and movement in the legs of the patients with Rett syndrome, but their sonoelastography, which is an ultrasound technique that measures muscle stiffness and elasticity, showed deterioration in the muscles. The second article, titled Use of a Low-Tech Tool in the Improvement of Social Interaction of Patients with Rett Syndrome, studied whether the addition of a low-tech tool called Click for All into cognitive and motor training of individuals with Rett syndrome helped increase social interaction. And the result was very positive. It resulted in improved social interactions and reduced distancing from peers. Under my week 9 post, a peer posted a link to an article that confirmed the use of trofinetide as a safe and effective treatment for Rett syndrome. This article helped me out a lot in my research since it gave me sort of a starting point to expand my knowledge on the subject. The clinical trial I read about the topic didn't post the results of the experiment, so it's also great to know the results are positive and strides are being made to making these individuals' lives a bit easier. Another one of my peers mentioned the potential of Rett syndrome being mistaken for autism spectrum disorder, also known as ASD, in young children, since some of the symptoms are so similar. This is a very valid concern, especially knowing how common ASD is compared to Rett syndrome. I am sure this misdiagnosis has been made in the past. Luckily, there are observable differences between the two. As a child with Rett syndrome grows older, the symptoms become more specific and refined. And of course, as research is advancing with Rett syndrome, the symptoms will also become more clear when compared to ASD. One final question I was asked under my week 9 post was, what is the role of MECP2 in adults? Which is a great question. I am pretty focused on children in this research that I've been doing. In the adult brain, MECP2 helps maintain connections between neurons, which play a huge role in cell-to-cell communication. MECP2 also is able to process and rearrange mRNA molecules, which in turn produce different versions of proteins known as alternative splicing, which is a process that also plays a large part in regulating neuron communication and the function of brain cells.